Hi everyone, thanks for tuning in. My name is Managopal, and I'm a researcher in the Englander Institute for Precision Medicine here at Weill Cornell. In this video, I'm going to demonstrate the process for standardizing a Tier 1 gene variant interpretation in the precision medicine knowledge base. I'll show you how to find a gene variant on the PMKB.org website, how to begin the revision process, how to consult additional resources for the most up-to-date information, and how to add citations in PMKB. Before we get started, I just want to say that the workflow presented here is meant to be illustrative of how to use external databases to acquire information that can be used in the interpretations that you write. It won't be the same for every variant, and you may find that you develop your own workflow that works better for you. Okay, with that being said, we can get started. So the first step of the process entails going onto the PMKB.org website, so navigate there in your browser. As you can see, we're here already. The second step entails logging in using the link at the top right corner of the page. Your login credentials will only be valid if you've registered your email address with us, so if you haven't done that, please do so and then resume at this step. As you can see, I'm already logged in. And now that we're logged in, we can find ourselves a variant to update and revise. We're going to do so by clicking on the Interpretations bar at the top. Okay, so now that we're here, we're going to select a variant interpretation to edit. As you can see, PMKB hosts a large number of interpretations already for actionable variants. I'm going to search for a variant of the MET gene to update. We can see we find one right here at the bottom. So I can click on the icon for the MET gene, and it will bring me to a page showing all the variants that have interpretations for the MET gene in PMKB. As you can see, these interpretations vary in format, content, and relevance. To review, these are the major bits of content that must be in an updated interpretation that you submit, and it must follow this structure. So I'll give you a moment to take a look at that. So as I said, this information is useful for crafting the parts of the interpretation pertaining to the molecular function. However, it's highly possible that this may be incomplete. So we'll check some other resources as well. But before I go, I need to make sure that I cite the proper reference for the information that I'm including in my draft. Now, I've already read through all of these references, and I know that the information that I pulled is from this source here. So, because I need the PubMed ID when I submit the interpretation on PMKB, I'll need to go to PubMed and find the PM ID. And here we see it at the bottom, so we can copy and paste this into our draft. Okay, so one great resource that we'll consult is explicitly recommended by the pathology department here at Weill. It's the paper DNA Sequencing of Cancer, What Have We Learned? By Julianne Chmielewicz and Matthew Meyerson. I'd highly suggest that each contributor read through this paper, if just to get a basic understanding of the landscape of cancer driver mutations. This review article describes and categorizes many of the most important cancer genes, and we can use it as a very effective resource. Of course, you should always use PubMed to find even further information that may be relevant to an interpretation. Anyway, looking for MET in the Chmielewicz paper, as we scroll down, tells us that they're activating mutations, and that it acts through the MAPK signaling cascade. And looking even further, we can see that MET has activating mutations that act through RAS and RAF to activate MAP kinase. So we can update our interpretation with this information. And if you consult the paper, make sure to include its PubMed ID in the citations. You can see it's right here. We'll add it to our list of references. Let's begin checking out some other resources. A few other great databases to check as part of your standard workflow are Cosmic, OncoKB, and the Jackson Labs Clinical Knowledge Base. These are excellent resources to use if you're trying to determine if the mutation that you're writing about is oncogenic or a hotspot mutation. First, let's check out COSMIC. We can search for MET. We can click on it. Now, as you can see, there's a great wealth of information, with the discussion of an overview of important facts, and further down, there are details on drug resistance. If we keep scrolling, there's even more information on the observed mutation rate across tissues. And most importantly, if we scroll all the way to the bottom, we see that there's a great list of primary references associated with the gene. In this example, we'll take a look to see if there's any detail on the mutation that we're interested in. So just to refresh, the variant that we're writing about is at amino acid position 1010. It's a D to H, N, or Y missense mutation in non-small cell lung cancer. So let's take a look at variants in position 1010. Now we can see that there are two variants here corresponding to position 1010, but they're not the ones that we're interested in. So it seems like what we're looking for isn't in COSMIC, but the rest of the information in COSMIC, especially the references, is very informative, even if only a fraction of that content makes it into an interpretation. Now let's check out OncoKB. Okay. And let's search for MET. As you can see, OncoKB gives us an informative and brief blurb here at the top. And after clicking on the little arrow for more information, we can read even more about the gene and how it's affected by mutations. Some of this information is unnecessary or redundant with our PMKB interpretation, but we'll read it anyway and look for anything that might be relevant. As I read through it, I see that the pathways affected by somatic mutations in MET also include PI3 kinase, and we can see that MEK is identified as a downstream molecule from RAS and RAF. Now, MEK is a specific type of MAP kinase, so that's even further granular detail that we can add to our interpretation, that we don't need to get too granular. Nonetheless, I think that that's something that we can include. Let's go back to our interpretation and modify this. Okay. Now we also have to make sure that we cite this information. We can see that OncoKB also includes the PMIDs of the resources that were used to generate this little blurb. I've already read through these references and I know that they apply to the parts that we just drew from the interpretation. So I'm going to copy and paste these. Since we're looking at codon 1010, we can scroll down to see if there are any explicit annotations of our variant in OncoKB. We can see here that all three of them are. If we click on one, we can see that it's been classified as likely oncogenic and likely gain of function. If we click on the others, we'll see the same thing, likely oncogenic and likely gain of function. Which would make sense considering that they're grouped together on PMKB. And so we can include this information in our ongoing interpretation draft, as that's part of the 4 points that constitute an interpretation. So we'll write the following. All right. Now, for any information that we draw from other databases, we'll need to cite the appropriate literature. In this case, OncoKB features the PMIDs of the articles relevant to the likely oncogenic nature of this variant. So I've already read through and I know which of these is relevant. So I'm going to copy the PMID. All right. There you go. So as I mentioned, another resource that we can check is the Jackson Labs Clinical Knowledge Base. So let's take a look at it. We can look for MET. Let's give it a second. We can see that it's right here. So the Jackson Lab CKB is a great resource to see if a mutation is oncogenic or a hotspot. The Jackson Lab has done a number of experiments where they've induced these mutations in cell lines to see if they're oncogenic. And that information is captured here in the database. Other resources that you can include are My Cancer Genome, and Civic. And of course, there's always the literature that you can find through PubMed. Now for many Tier 3 variants, which we're not really covering in this video, the variants may be germline, and so a great resource to look at is ClinVar. Okay. Next, we can start looking at the information regarding prevalence and prognostic and therapeutic significance for these gene variants. If we head back to OncoKB, we go to the main page for this gene. We can hover our mouse over here, and it shows us the frequency of MET mutations in the MSK-CC cohort. And we can see that it's about 3.2% in non-small cell lung cancers at MSK. And for the variants themselves, we see that they range in between around 0.2% to around 0.6%. And since we're going to be citing this paper, let's make sure that we have its PubMed ID. All right, that's not it. Right here. Okay. Okay. Now for prevalence information, we can check out another another resource. It's called CBio Portal. And CBio Portal is a great resource that hosts summary statistics for a number of large cancer sequencing studies. We'll scroll down to the cohort that is relevant to the cancer that we're writing our interpretation for. So we're going to go to lung and look for non-small cell lung cancer. Here we go. We'll focus on the largest cohorts that they have. We'll focus on the MSK-CC cohort and the latest TCGA cohort. We can scroll down and type in the gene that we're interested in, which is MET. We can submit our query. Now here under the Cancer Type Summary tab, we can see the frequency that MET was altered in these cohorts. So we can hover our mouse over these stacked bar plots. And we can see that overall, in the MSK cohort, MET was altered around 7.5%. And more specifically, it was mutated around 5.4%. For the TCGA study, we can see that overall it was altered in about 4.9% and mutated in 2.9%. In our case, because we're considering mutations, in particular missense mutations, we're going to be going with the latter numbers, the 2.97% here and the 5.4% here. Now, if we go to another tab, go to Mutations, we hover over here, we can see that the somatic mutation frequency overall is around 3.5%, and that's across both studies. So this is the number that we're going to use for overall alterations of the gene for our interpretation. So we can start including this information from CBio Portal and the information that we got from OncoKB and start drafting that into our interp. So right now, we'll write the following. So MET mutations occur in 3.5% of non-small cell lung cancer cases overall. And this last line right here is specifically for this type of variant, which we pulled from OncoKB. Okay, so since we pulled information from studies hosted on CBio Portal, we have to make sure that we cite them. So we'll go back over here and pull the PubMed IDs. So we'll scroll back down and we'll find the references for those cohorts. We can find them right here. Again, it's a little book icon. So we can open this one up and this one up in a new tab. And we'll pull their PubMed IDs. So this is for the MSK study. And this is for the TCGA study. All right. So now we've covered 3 out of the 4 points for an interpretation in PMKB. We've got the biological function, a description of the variant's role in oncogenesis, and a description of the prevalence of the mutations. Lastly, we want to see if there's any therapeutic or prognostic significance to MET mutations in non-small cell lung cancer. So let's check back to OncoKB and see if there's anything listed. So, as we can see, it looks like for non-small cell lung cancer, the drug crizotinib is suggested. And this is level 2A evidence, which means that this set of MET variants is a biomarker that's predictive of response to crizotinib, which is an FDA-approved drug. And this also corroborates the information already present in the pre-existing interpretation for this variant in PMKB. And true to form, OncoKB also lists the annotations, the references that are associated with this drug indication, and we can make note of those PMIDs. All right, so now let's copy and paste these PMIDs into our existing interpretation draft. So all right. So now, why don't we check out CBio Portal, because that also may have information regarding treatment. So we head back to the Mutations tab. We can look across the spectrum of variants that exist here. And we can see that actually, the mutation that we're interested in is right here. We can click on it. We can scroll down to see more. We can see for this particular mutation, the D1010N, there are annotations that exist. And this is actually pulled from OncoKB. So it's more or less the same information that we were just looking at, but it gives us a little more. And it tells us some more specific information about the indication for crizotinib in non-small cell lung cancer with MET mutations. And it tells us in particular that MET exon 14 alterations, such as D1010N, and therefore our other variants, are an indication for crizotinib. So we can make note of that in our running interpretation. So we can write okay, and now we've pulled up all the necessary information for an interpretation in PMKB. In the event that we couldn't find this in OncoKB or CBio Portal or Civic or the JAX-CKB, we could have always searched for the literature on PubMed. And other than PubMed, we can also look at the NCCN guidelines. Let's take a look. And so the compendia here, especially for biomarkers and then also for drugs and biologics, are really useful. And otherwise, you could also look up FDA drugs to see if there are any indications for the gene variants that we may be writing interpretations for. So we can go to any of these links, and that'll give us more information. So we've now drafted a complete interpretation covering all necessary points. The last part is to organize our interp into the necessary format, collect all of our PMIDs, and submit it for review. So if you remember, the final interpretation will feature the prevalence and prognostic or therapeutic information in the first one-liner. So we have to reorganize our interpretation as it's been drafted. So we can reorganize it as such. Okay, and now we can post this on the PMKB website. Let's copy this, and we'll go to our interp. Let's click Edit, and let's scroll down. All right, and we'll add our references. Okay, and now we can submit. Just adding a final versioning comment, and we'll update the interpretation. And it has been submitted for review. Okay, so now the next step is absolutely necessary if you're contributing to the crowdsourced competition. The next thing that we need to do is log our newly submitted interpretation in the Google form for tracking submissions in the competition. So we'll copy this link here from the PMKB website and log it into the Google form. We can pull it up here. As you can see, it's very simple. All we're going to do is copy and paste that link right here, and we're going to log that it is a new edit, and we'll submit. So after submitting on the Google form, we'll make sure to check back on the Google Sheet that's associated with it. There will be periodic updates with comments from reviewers. As you can see, there will be a date associated in the comments and the interpretation status. Basically, if it's pending or if it's been approved as an interpretation on the PMKB website. Here in the comments, you'll be seeing the edits and revisions that the pathologists associated with this study will be making in order to move your interpretation along to getting approved. From there, you'll also be able to see reviewer comments on other contributors' submissions, and you can learn how to make even stronger interpretations for yourself. Okay, so in this video, we've walked through how to find an interpretation to edit on PMKB, how to acquire the information necessary to draft an interpretation, and how to acquire the appropriate references. This video tutorial was meant to be illustrative of how to make use of other knowledge bases for acquiring the information relevant to the gene variants that we're writing about. I just want to emphasize, it won't be the same for every variant. For many variants, there may not be a targeted therapy, or the variant may actually confer resistance to therapy. Or the variant may simply have an association with overall survival, but without therapeutic significance. And as for drafting other interpretations, you may find that the external databases are less useful than just going straight to the primary literature. Always, always, always make sure to actually read the articles that you're citing and ensure that the information is actually there. And that's it. Thank you guys so much for watching. Please feel free to reach out to me with any questions.